Okie doke.
So this is Esther Helga again, Saturday, December 8th. And we pleasantly introduce Robert H. Lustig, MD, an internationally renowned pediatric, and here comes the word that I've been practicing so I can say it, endocrinologist. Yeah, well.
Very good. Very good.
Who has spent the past years treating childhood obesity and studying the effects of sugar on the central nervous system, metabolism, and this is disease. He's director of different organizations, and for me, you know, such an inspiration on sugar and the harm it does in the world. And I've just been so personally appreciative of your work at going out there and putting your force, which is incredible, into this field for all of us. So it's a great honor for me to present you here today.
Well, thank you, Helga, and thank you all for spending Saturday, you know, learning about this issue. I'm very happy to, you know, sort of give you my take on this whole question and issue of food addiction. I know that Nicola Vina presented to you 2 weeks ago. I was on that webinar as well. And what I'll try to do is sort of pick up where she left off. I'm going to do my absolute level best not to be redundant with her, but rather be complementary with what she has told you, so as not to waste your time, and also not, so as not to confuse you. The field is confusing enough as it is. And you can see from the title of the talk, both for and against this concept of food addiction, and you will see why that is. And it has a lot to do with the question of what is food.
Did you ever get the driver's license?
Brought that up. We're gonna dive deeper into that question today. All right. So first of all, let me go ahead and put it on presentation mode now. All right. So I do have a couple of disclosures. I did write these 2 books for the general public: Fat Chance, which is about diet and physical health, and also more recently, The Hacking of the American Mind, which is about diet and behavioral health, and in particular, the issues of addiction and depression, and the role that food and other things like drugs and technology play in this current pandemic of addiction and depression that we currently find ourselves in. I'm also the chief science officer of a nonprofit located here in the Bay Area called Eat Real, where we are committed to the reversal of childhood obesity and type 2 diabetes. We certify restaurants that procure, market, and sell real food. We teach 4th graders how to cook. And the goal is to try to start a public movement around the concept of real food in an attempt to try to influence policymakers, the food industry, and governments. And then lastly, I am the chief medical officer of a for-profit startup called Biolumin Technologies, because we unfortunately recognize that despite our best efforts, processed food is not going away. And to be honest with you, I'm not even sure we really want it to go away, because we have to feed 9 billion people by the year 2050. And we can't do it with the current land and water use that we have available to us, which will only get worse. So we actually have to find ways to decrease depreciation. Yet, at the same time, we have to mitigate the metabolic risk associated with processed food. We have to find a peaceful coexistence with it. And we are trying to bioengineer an alternative to our current food pandemic that will allow for both health and economics. And that's what, but we will not be discussing any of those things on the call today. All right, you've seen this slide, I'm sure, already. Junk food addiction may be clue to obesity, indeed. there's plenty of data on the notion that junk food has addictive potential, and we're going to go through some of that today. Nicole, of course, did a lot of that just 2 weeks ago. So, is food addictive? The lay public seems to know, and there are all these books out there talking about this concept of either food addiction, sugar addiction, etc. And the question is, what's the real science behind this? And what do we know, not just in animals, but in humans? So, in humans, we now have this metric called the Yale Food Addiction Scale, which has been validated by Ashley Gearhart, working with Kelly Brownell at Yale, and she's now at the University of Michigan. And these are the kinds of questions they ask on this questionnaire for this scale. And it basically works using the DSM-4 criteria. But as you know, we're now up to the DSM-5 criteria. And we're going to talk about the difference between those two as we go as well. So in a sense, this Yale Food Addiction Scale is somewhat outdated because it's using a different DSM set of criteria. Nonetheless, this food addiction scale does correlate with neuroimaging. If you're looking in the anterior cingulate cortex, as this does, you can see that food addiction scores correlate with activation by fMRI in this particular area, and also in other areas, including the nucleus accumbens, which is, of course, the reward center, which you're all very familiar as addiction specialists with. And palatable food cues trigger these areas specifically, not just food addiction, but when you give those specific palatable foods, you can see changes in BMI commensurate with the activation of these particular areas, suggesting that at least obesity is one readout of the addiction pathway. Well, this has led to this concept of food addiction, that there is something about food that can be addictive. And as you'll note, Mark Gold, who is one of the pioneers of this, he was the chairman of the Department of Psychiatry at the University of Florida, was probably the main mover in getting this into the medical literature. And here are two supplements to two journals that Mark edited, both about the neurobiology of food addiction. What's interesting is that here in 2009 and 2010, he called it as he saw it. He called it food addiction. Yet, in 2012, all of a sudden it changed from food addiction now to food and addiction. They're not the same. Food addiction is about the food. Food and addiction can be about anything you damn well please. And the question is, here's Mark Gold, saying two different things in, 2 years apart. So the question is it that food is a manifestation of addiction, or is it that food is a cause of addiction? And this has, sort of occupied an outsized portion of this addiction literature for the past 7 to 8 years. So much so that this group in England, of which Sadaf Faruqi, who's an obesity geneticist whom I know well, along with these two psychiatrists, Ziaduddin and Fletcher, came up with this opinion article.
Rob just sent me, he's giving a live talk in Iceland about food addiction.
Say again? Couldn't hear? Hello? Cannot hear. Am I supposed to be able to hear?
No,
Okay.
it was somebody just dropped in.
Oh, okay. Oh, I asked my colleague Laurel Mellon if she would join, and she has. I'm so pleased. I'm very pleased about that. So anyway, in 2012, like I said, this group in England asked the question, is it food addiction or is it not? In Nature Reviews, to which Nicole and Ashley and Mark wrote this response 2 months later, tossing the baby out with the bathwater after a brief rinse, the potential downside of dismissing food addiction based on limited data, to which then The British group responded back, food addiction, is there a baby in the bathwater? So as you can see, this question of food addiction versus food and addiction continues to generate heat rather than light. And my goal in today's talk is to try to generate some light and explain to you why this controversy exists, what the specifics are, how the science can inform this question, and what you should be able to take away as addiction specialists going forward in order to be able to help patients and hopefully help the field. So, as we've just discussed, there's this group in Europe. They're called NeuroFast. Anybody familiar with NeuroFast? Maybe some of you even belong to NeuroFast, for all I know. We're going to discuss why they say this. They say that their research says that there is this thing called eating addiction, and that is very different from food addiction. And while there is an addictive-like eating behavior, that it's not the food, that it's the person. So NeuroFast puts the onus on the individual rather than the food. Now, if it's about the individual, then we can't expect any public policy help going forward. If it's about the food, then we absolutely need public policy help to try to deal with the problem. So this is not a semantic issue. This is a public policy issue. So if it's about obesity, or if it's about eating addiction, or if no specific food stuff is addictive, then in my view, at least, the food industry will have carte blanche, and they have had carte blanche, and they will continue to have carte blanche because there's no option for societal intervention. So answering this question, food addiction, eating addiction, is absolutely essential. It's seminal to where we need to go. So let's look at the NeuroFast website and find out what it is that they say and why they say it. So this is a direct screenshot from their own website. What they say, here are the there are 4 points to make. Here's the first two. Current evidence does not allow us to conclude that a single food substance via a single specific neurobiological mechanism, that is brain receptors or neural pathways, can account for the fact that people overeat and develop obesity. Now, I want you to look at that statement very clearly, okay? Single food, obesity. What you will see consistently through this entire discussion is that obesity is their benchmark for addiction. I think this is incorrect. I think this is the mistake. Let's go on. In humans, there's no evidence that a specific food ingredient, or food additive causes a substance-based type of addiction. The only currently known exception is caffeine via specific mechanisms, which can potentially be addictive. So they recognize that caffeine, which is in food, can be addictive. Okay, so keep that in mind because we're going to return to that in a minute. Next, within this context, we specifically point out that we do not consider alcoholic beverages as food, despite the fact that 1 gram of ethanol has an energy density of 7 kcal. So here's an energy source that is calories that they exclude from food. Now, I understand why they do that, and I don't think there's a dietitian on the planet that would argue that alcohol is food, but it is an energy source, and it's clearly addictive. So caffeine and alcohol become the template and the discussion point for the rest of this evaluation and the rest of this legal argument as we go. And lastly, addictive overeating is clearly distinct from substance use disorders that cause addiction via specific mechanisms. That is nicotine, cocaine, cannabinoids, opioids, etc. And the reason is because, well, food is not drugs in their view. So you'll notice that NeuroFast exempts both alcohol and caffeine. Yet both of those are found in food. You could say, well, alcohol is not found in food, but yes, it is, because anytime bacteria work on a piece of fruit on the vine, it's making alcohol. There's alcohol, and it's one of the reasons why animals during harvest time get drunk out in the field, because they're consuming the fermented fruit with the alcohol in it. All right, now to understand the science, we have to understand the neural pathways that are involved. And this is where I've done some of the work, and why I feel comfortable and confident in discussing these issues with you. So this slide here is the neuroendocrinology of energy balance in one slide. What you'll notice is that there are 4 hormones: leptin, insulin, ghrelin, and peptide YY, and they're the ones with the blue arrows. Now, they're coming from peripheral organs, and they're all impacting on the brain, in particular the hypothalamus, in particular the ventromedial hypothalamus, as you can see. Now, ghrelin and peptide YY are alimentary hormones. They're hormones that are created and released meal to meal. They regulate the peripheral alimentary system response to food, telling the brain where you are in relation to a meal. Now, when your stomach is empty, ghrelin goes up, and ghrelin tells the brain, hey, I'm hungry, feed me. So you eat, and then ghrelin goes down. So ghrelin is a stimulus for hunger. So by induction, you would then expect that a ghrelin suppression would be the end of the meal. You would be wrong. Ghrelin suppression after you consume food is the end of hunger. It is not the end of the meal. Everyone can eat past the end of hunger, and do, routinely. And the reason is because ghrelin going down is not the satiety signal. Ghrelin going down is the end of hunger signal, but the end of hunger and satiety are not synonyms. In fact, there's 20 minutes between the end of hunger and satiety. And the satiety is mediated by this other hormone called peptide YY3-36. Now, it's made in the distal and small intestine, in the ileum, in the same cells that make glucagon-like peptide 1 called L cells. When the food finally gets to the end of the small intestine, peptide YY is released into the bloodstream, travels through the bloodstream to the brain, binds to receptors in the ventromedial hypothalamus called Y2 receptors, and that tells the brain, the meal is over. I'll die before I eat again. That is that is the satiety signal. So the end of hunger comes when you put food in the stomach. Satiety doesn't occur until the food makes its way through the intestine. There's a 20-minute delay between those two phenomena, which is one of the reasons why in our clinic we make one of the rules: wait 20 minutes before second portions. Give the hormones a chance to work for you instead of against you. But these two hormones, ghrelin and peptide YY, these are acute hormones. They're minute-to-minute hormones. They're meal-to-meal hormones. They don't tell your brain what's going on long-term. That's where these other two hormones come in, leptin and insulin. And that's what we're going to talk about for the rest of the morning. These two hormones, leptin and insulin. Now you'll notice that leptin comes from the adipocyte here, enters the bloodstream, goes to the brain, and tells the hypothalamus, hey, I've got enough energy on board to not eat everything in sight and to engage in normal, expensive metabolic processes, such as puberty, such as pregnancy. If you are leptin deficient, if your adipocytes are not making leptin, you don't go into puberty because your brain thinks you're starving. You can't get pregnant or can't maintain the pregnancy because your brain says, I can't afford the energy that it would take to grow a fetus. So this is a long-term metabolic adiposity signal that tells your brain you have enough energy on board so that you don't have to eat everything you see. So leptin is an adiposity signal, not a satiety signal, not a hunger signal, but an adiposity signal. Everybody understand that and understand the difference? Now, insulin is my favorite hormone. And the reason insulin is my favorite hormone is because you'll notice that there are two arrows coming out of insulin, and they're going to two different organs. One arrow is going from insulin to the adipocyte, because insulin is the energy storage hormone. Insulin opens the key, is the key that opens the lock to the adipocyte that lets energy rush in. There is no weight gain without insulin. There is no energy deposition without insulin. Insulin drives weight gain. Insulin drives energy storage at the level of the adipocyte, always. Now, you'll notice that there's another blue arrow coming out of insulin, and it's going to the brain. What does it tell the brain? It tells the brain, hey, I'm in the middle of metabolizing a meal. I don't need any more energy right now. I got to work on what I'm working on right now. And so it tells the brain, stop. So it tells the fat cell store, and it tells the brain, stop. Two separate, dichotomous, in opposite direction signals. Store, stop. And that's why insulin is the linchpin in this entire story. Because when insulin doesn't tell the brain, stop, you keep eating. And we now know what insulin's doing at the level of the brain to keep it from telling the brain, stop. And it is that phenomenon that drives food addiction. So understanding how insulin goes from a part of the satiety signal to part of the starvation signal is what this whole field of obesity is about. So let me try to explain that to you in this slide. This is a concept that we, my colleague Michelle Mita-Snyder and I came up with now 10 years ago, which we call the limbic triangle, kind of like the Bermuda Triangle. Once you get in, you can't get out. Same thing here. So you see obesity here at the bottom, right? And so when you store energy, you increase weight gain, you increase adiposity. Well, those adipocytes then make leptin, and then the leptin will act on two separate areas of the brain. The first area it will act on is this hypothalamus we've been talking about. When leptin tells the hypothalamus that things are working, you get the adiposity signal, you can eat less. But when leptin is either deficient or whether something's blocking that leptin signal, you end up with this phenomenon called leptin resistance. Leptin resistance is brain starvation because your brain thinks there's no leptin. And if your brain thinks there's no leptin, your brain thinks you're starving. So if leptin can't signal, then you're going to keep eating because your brain thinks you need more energy. Turns out the cause of that leptin resistance is that hormone insulin. Insulin blocks leptin signaling at the level of the hypothalamus through specific molecular pathways that have now been delineated by various labs around the world. Marty Myers, Jens Bruning, Ron Kahn, several others. Turns out that the leptin receptor and the insulin receptor co-localize, and it turns out that when you turn insulin on, you end up turning leptin signaling off. And I'll show you that on the next slides. In addition, there's this second area of the brain over here called the ventral tegmental area. And that should be very well known to you since you are all addiction clinicians and specialists and researchers. So that's where the dopamine cell bodies are. And they ultimately synapse on the nucleus accumbens, which is the reward center. And that's where dopamine signal transduction occurs. Well, it turns out those VTA neurons also have insulin and leptin receptors. And insulin blocks leptin signaling there as well. And so when insulin blocks that leptin signaling, you can't extinguish the dopamine signal, which means you end up heightening your reward. Then throw the third vertex of the limbic triangle on top, which is the amygdala. And as you know, the amygdala is your fear center, your stress center, your cortisol generation center. So stress generates cortisol. Cortisol specifically induces insulin resistance at the level of the visceral tissue, visceral adipose tissue and the liver. And cortisol is also a direct stimulator of appetite, in particular, high energy density food, high carbohydrate, high fat, high fructose, all of which also lead to insulin resistance. So you can see that when you're stressed, you're going to increase the value of food as reward. You're going to increase your insulin resistance, which will therefore increase leptin resistance, decrease leptin signaling, therefore make you think you're starving, make you need more reward, and drive this limbic triangle into a vicious cycle. So these are the three areas, these are the three hormones involved in this pathway: leptin, insulin, cortisol. All right, so I mentioned this insulin blocking leptin signaling. Let's show you how that works. So here's your nucleus accumbens right here. Everybody sees the arrow, right? Where I'm pointing? Yes, because I'm sharing the screen. Okay, so the hypothalamus communicates with the nucleus accumbens. Here's the leptin receptor, but there's a leptin receptor here on the VTA as well. And they're both feeding the nucleus accumbens. This is where reward integration occurs. And it's taking inhibitory information from the prefrontal cortex, which would normally tamp down on reward-seeking behavior. The hippocampus, which is the memory center, which also tamps down on reward-seeking behavior. And then, of course, there's the amygdala, which is where the fear center is, which drives the cortisol. All of these are impacting on the nucleus accumbens and modulating how well leptin is working to turn this off. Let me give you the paradox that explains this so that everyone understands. If you give a 5-year-old kid a cookie, what happens? They bounce off walls. Okay, it's called the sugar high. And the reason this happens is because the insulin drove the energy into the adipocyte. The adipocyte made leptin. The leptin circulates, goes to the hypothalamus. The hypothalamus, recognizing that energy has come on board, will activate the sympathetic nervous system, increasing sympathetic tone to the level of the muscles, which cause fidgeting in an attempt to try to burn it off in order to return the adipocyte to its previous size and mass. And so this is a negative feedback pathway between the fat cell, the brain, and the autonomic nervous system to keep you in energy balance. And it works as long as leptin works. Now, here's the paradox. What if you give a 5-year-old obese kid a cookie instead of a normal weight 5-year-old kid a cookie? They're in the pantry looking for more cookies. Anybody ever see a sugar high in a fat kid? Doesn't exist. Does not exist. Thin kids get sugar highs. Fat kids don't. And that's the point. Because the fat kid is insulin resistant and leptin resistant. And we can show that. So here's phosphostat 3 in mice, in the hypothalamus and in the ventral tegmental area. And you can see that leptin ups the phosphostat 3 greater than vehicle. And you can see it's true in the reward center, the VTA, and you can see it's true in the hypothalamus as well, the medial basal hypothalamus. Okay, so far so good. But what you'll notice in this study what was done here was these mice were given an RNA interference. In other words, a molecule of RNA injected right into the VTA, right where the dopamine neurons are, which knocks down the leptin receptor. So there are fewer leptin receptors. So these animals are now functionally and genetically leptin resistant because their leptin receptors have been interfered with. So they can't see their leptin signal because they don't have the leptin receptor to transduce the signal. And when you do that, as you can see here, what happens? They start consuming what? Sugar. So the lack of leptin signaling is a drive to increase sugar consumption. In addition, leptin, this is work from Sadaf Farooqi in Cambridge, one of the people who wrote that editorial. What you can see is that in a normal person, when you give them leptin, the activation of the VTA and the nucleus accumbens is normal, but in the leptin deficient state, it's way high. And when you give leptin to people who are leptin deficient, you can make it go down because now their brain sees the leptin. In other words, you extinguish the addictive property of food by giving leptin in a leptin deficient person. And the reason is because both insulin and leptin receptors co-localize 100% in this area of the ventral tegmental area. These neurons very specifically transduce both insulin and leptin signals. When you give insulin, you increase dopamine clearance, meaning you're getting rid of dopamine when the cells are sensitive. Here's another example. You inject insulin into the ventral tegmental area, nothing happens. But if you gave an opioid, which would increase food intake here on the y-axis, you increase food intake. But if you give insulin with the opioid, you've mitigated the effect. So in every single case, when the brain is insulin sensitive, insulin stops food intake. Insulin says stop, as long as the brain is insulin sensitive. But what if the brain is insulin resistant? What if the brain can't transduce that signal? And that's this mouse, known as the NIRCO mouse, the brain insulin receptor knockout mouse of Ron Kahn. So in this mouse, they have taken the insulin receptor out of neurons. And what happens is their leptin goes up because now they're eating like crazy. Their body weight goes up because they're eating like crazy because they can't see their fat, because their insulin isn't generating a sensitivity signal. Their white adipose tissue goes up, their body weight goes up, their leptin goes up. These animals are fat because they can't get the signal from the periphery. And here's the pathway in those neurons. So these are the neurons in the hypothalamus and in the ventral tegmental area. Here's the insulin receptor. Here's the leptin receptor. They're different. The insulin receptor is a heterotetramer. The leptin receptor is a dimer. This is a cytokine receptor. This is its own receptor. What you'll notice is that there is this bridge between the two. These two signal transduction mechanisms here, SH2B and JAK2, coordinate with each other. If you take JAK2 out, you get a fat mouse. If you take STAT3 out, you get a fat mouse. But it turns out you can take the insulin side out too and get a fat mouse. Here's what's going on in the brain, in that area, okay? In the hypothalamus, not in the ventral tegmental area, but it doesn't matter. Look at this bottom slide down here. You're giving insulin, and you'll notice firing, 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 right? Sorry, take it back. It's wait. I'm trying to remember how this works. Here, okay. before you give the insulin, it's firing like crazy because the leptin's working. You give the insulin and now the neuron goes to sleep. That's it. You give the insulin and the neuron goes to sleep. So basically what's happening is the insulin's blocking that neuron from firing when you give it chronically. And if you take cells that normally don't have either insulin receptors or leptin receptors and you transfect the cells with both of them, then you add insulin and now you end up with a reduced leptin signal. So in every one of these cases, adding insulin leads to lower leptin signaling, thereby increasing brain starvation, increasing the feeling of the need for reward. And we can show in humans that this is true as well. This is correlational data rather than causational data, but nonetheless shows the effect. So this is work that my friends, Joan Hahn, who's now at University of Tennessee, Memphis, took my job there, and Jack Yanofsky at the NICHD did. What they did, which was really cool, they took a bunch of fat kids, they drew their blood baseline in the morning, and then they let them loose at a metabolic buffet. So they could eat anything they wanted. There were 10,000 calories on that metabolic buffet in different kinds of foods. And there was a little scale underneath each plate. So they could measure everything that was taken. And they were able to then determine what the energy intake at that metabolic breakfast was. And it turned out that the higher the log first phase insulin and the higher the HOMA, which is a measure of insulin resistance, the more energy they took in. And this is clearly cause rather than effect because it's happening on the front end because they drew the blood beforehand. So the higher the insulin, the more food you eat. You would think that if insulin said stop, the higher the insulin, the less food you would eat. No, because your brain is insulin resistant. So chronic hyperinsulinemia is the driver. It drives energy into adipose tissue peripherally. It interferes with leptin signaling in the hypothalamus, which we call brain starvation. And lastly, it interferes with leptin's ability to extinguish dopamine clearance in the nucleus accumbens. And that's the addiction part. That's the indirect part of addiction. So insulin is the bad guy. The goal is get the insulin down so that your leptin can work. So back to the limbic triangle. Here's obesity. Obesity is actually the manifestation of the hyperinsulinemia. The insulin comes first. The insulin is the bad guy. Now, there are two phenomena that raise your insulin. One's called insulin secretion, that is the pancreas making insulin, and the other one's called insulin resistance, your liver not responding to insulin. Both will raise your insulin. When your insulin goes up, your fat goes up. When your fat goes up, your leptin goes up. Now, your leptin should turn this off, but it doesn't because of leptin resistance generated by the insulin. When the leptin goes up, it should turn starvation off, but it doesn't because of the leptin resistance generated by insulin. Insulin blocks leptin signaling. Everybody got that? So, of course, the question is why? Why does insulin block leptin signaling? What is the evolutionary construct from a Darwin standpoint? Why would insulin block leptin? Anybody? Can anybody think of the reason that God would make this so? We mentioned it earlier.
So
Go
that
ahead.
you can carb load and then and not starve to death?
So that you could what and not starve to death?
So that you can eat a lot of carbohydrate and keep that in your tissue and your fat so that you don't starve to death in famine?
Nope. That could be that. You wouldn't need it for that. I'll tell you why you need it.
Good.
I'll do it. There are two times in your life where you want leptin to not work. Okay? If leptin worked all the time, you could never gain the weight. There are two times in your life you actually have to gain weight. And we mentioned them earlier. puberty and pregnancy. If you don't gain weight during puberty, species dies out. If you don't gain weight during pregnancy, species dies out. If leptin worked right all the time, species dies out. There has to be some modification of the leptin signaling pathway to allow for puberty and pregnancy so the species doesn't die out. And it turns out that insulin is that pathway. That's the modifier. And it makes sense that that should be the case because after all, shouldn't the hormone that causes the weight gain peripherally be the same hormone that interferes with the stoppage of food intake at the central level? That they would be yoked together. So insulin blocks leptin. And leptin is a good guy. And insulin, when we're talking about obesity and addiction, is a bad guy. Everybody with me so far? Okay, that's important because then you have to ask the question, okay, what are the insulinogenic foods? And that's when we get into the food question. The direct effects on the reward system, not the indirect effects, but the direct effects. What makes dopamine go up? Okay, so there are a bunch of things that have direct effects on the reward system, but dopamine is the most important. Yes, opioids are important. Acetylcholine is important. Stress is important. But we're just going to talk about dopamine today because we only have so much time. All right, so everybody's familiar with the dopamine system now, right? The mesolimbic dopamine system. Here's the ventral tegmental area. Here's the dopamine pericardia. Here's the nucleus accumbens. The amygdala is here. The prefrontal cortex is here. The hippocampus is here. The locus ceruleus, which is the adrenergic center, is over here. They're all communicating with each other. But this is the primary driver of the system, VTA to nucleus accumbens. And this is the dopamine neuron here. Okay. Now, you're all familiar with cigarettes. You're all familiar with nicotine. How does nicotine work? Well, what nicotine does is it blocks GABA. It's a GABA antagonist. By being a GABA antagonist, you basically disinhibit that dopamine neuron. And so here it is going, here's that dopamine neuron from the VTA to the nucleus accumbens going bing, bing, bing. You throw some nicotine on and now go bing, 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 because you've blocked the inhibitory pathway of GABA on that dopamine neuron. Everybody got that? Okay. Well, turns out opioids are part of this as well. So here's the injection of opioids into the nucleus accumbens directly, and you can see food intake is a readout of that effect. So when you stimulate dopamine at the level of the nucleus accumbens, and it can be by opiate or nicotine, you will increase food intake. Well, here are humans. Here's that nucleus accumbens. We're lighting it up with C11 raclopride. So we're basically doing PET scanning for those dopamine receptors, D2 receptors in the nucleus accumbens. Here's a controlled brain. And here is a cocaine addicted brain. And you'll notice that the D2 receptors are way down. They have been downregulated because dopamine downregulates its own receptor. Now, the question is, why does dopamine downregulate its own receptor? And the answer to that is because dopamine is an excitatory neurotransmitter. Dopamine excites the next neuron. Whenever dopamine is released, the next neuron fires. Now, neurons like to be excited. That's why they have receptors in the first place. But neurons like to be tickled, not bludgeoned. They like to be stimulated, then they like to go back to rest. Chronic overstimulation of any neuron, anywhere in the brain, will lead to neuronal cell death. And we know that because of kids with chronic seizure disorders that I take care of as a pediatrician. They end up in the intensive care unit. Sometimes we have to put them into a coma in order to stop those excitatory neurons from stimulating the next neuron. because the more they stimulate them, the more neurons die, and the more retarded the kid ends up if the kid doesn't die from it. All right? So chronic overstimulation of any neuron will lead to neuronal cell death. So the receiving neuron, the postsynaptic neuron, it has a plan B. It has a self-defense mechanism. It has a method of self-preservation. What it does is it downregulates the number of receptors. So there's less likelihood that any given molecule of dopamine will find a receptor to fit into. So you can reduce the dopamine or you can reduce the receptors. Either way, you will reduce the gain on the system. So if the dopamine's coming fast and furious, what you can do is withdraw the receptors in an attempt to try to preserve the neuron. And that's what's going on here in this cocaine brain. This downregulation of the dopamine receptor. So in human terms, you get a hit, you get a rush, receptors go down. Next time, you need a bigger hit to get the same rush because there are fewer receptors. And then the receptors go down. And then you need a bigger hit and a bigger hit and a bigger hit until finally you get a huge hit to get nothing. That's called tolerance. And when the neurons actually do start to die, that's called addiction. Everybody got that? That's the end common pathway of addiction, is chronic overstimulation by dopamine of these dopamine receptor containing neurons that can't take it because they're fragile. So it can happen with cocaine. Here's the dopamine D2 receptor downregulation of cocaine. Well, here's obesity. Look, same thing. Everybody got it? The same downregulation of those D2 receptors that you see over here. Maybe not quite as bad with this particular slide, but you get the picture. It's the same phenomenon. And it's true in humans. As you can see, here is a BMI of 23, and here's a BMI of 50. Everybody recognize David over here? That's because he took a trip to America, right? Okay. You don't have that in Iceland, do you? I've been to Iceland. You have great lamb and fish.
Yes,
Love
and a lot of obesity for sure.
Yeah, well, yes, you do have a lot of obesity, but not so much ill health, which we can talk about later.
Yeah.
Monkeys, here's a thin monkey, here's a fat monkey. Again, you can see the downregulation. Here's a normal weight rat, here's an obese rat. Same thing each time, okay? As the obesity goes up, the dopamine receptors are going down because the goal is to protect the neuron. Well, what if you have a problem with your dopamine receptors in the first place? So it turns out people who have a tendency toward weight gain show a reduction in the striatal response to sweet versus women who were weight stable. What we don't know is if that's cause or effect. But we do know that there are genetic polymorphisms in that dopamine receptor gene, this one called the TAC1A allele. If you harbor this polymorphism, you have 30 to 40% fewer dopamine receptors to start with. What that means is you have to release more dopamine to get the same level of reward, which means you have to eat more. And so people who have this TAC1A allelic variation that's associated with low dopamine receptors to start with, they gain more weight over time because they need more food to get a reward response, because they have fewer receptors, because they already have a genetic downregulation of their receptors. They're already in the middle of tolerance because they have fewer receptors to start with. So there are some people who are genetically predisposed. And as you can see here, here's the weight gain with the A1 receptor versus not. And you can see it's much, the weight change over a 1-year period is much greater in those people with the 1A receptor. Okay, So that then leads us to the question of, okay, well, what stimulates the dopamine in the first place? So now we come to this question of neurofast. Is it food addiction or is it eating addiction? If it's eating addiction, then no specific food should be a problem. It's just the person. But if it's food addiction, then there should be specific foods that drive this. So, is fast food addictive? Michael Moss, you're probably familiar with, a Pulitzer Prize-winning New York Times reporter, wrote this book in 2013 called Salt, Sugar, Fat, How the Food Giants Hooked Us, okay? Addiction, right? He missed something. What did he miss? Salt, sugar, fat, and caffeine. Now we've got fast food. Salt, sugar, fat, and caffeine. That's fast food. Let's do each of them. Let's look at each one of these. Let's start with salt. Is salt addictive? Yes or no? What do you think? Just nod your head or shake your head. Yes, addictive. No, not addictive. What do you think? I see some nods. Okay. In animals, there is some data that suggests that there is a dopamine signaling response to salt, and that it could cross-sensitize with amphetamines in animals, and that would suggest that there might be an addictive component, as Nicole talked about 2 weeks ago. However, in humans, we know that there is a physiologically fixed threshold that you can actually retrain people to a lower salt preference, like the DASH diet, and that they will ultimately respond well to that. But it takes time, but you can, wean people off salt. And lastly, most importantly, as a pediatric endocrinologist, I have patients with this disease called salt-losing congenital adrenal hyperplasia. They can't make aldosterone because they have a genetic defect in their adrenal glands, which prevent that salt-retaining hormone from being made. And so these kids are salt cravers like you can't believe. They drink the pickle brine out of the jar, okay? They pour the entire salt shaker on their French fries, okay? They are the ultimate salt cravers. However, when you treat their congenital adrenal hyperplasia, when you get their salt-retaining hormone up, it goes away. If salt were addictive, it wouldn't go away. So, in humans, salt, not addictive. Next, fat. Is fat addictive? Yes or no? What do you think? Well, rodents do binge on fat, no argument, but they only binge on fat in the presence of sugar. It's called cookie dough. So whenever you see a study done in rats with a high-fat diet, it's really not a high-fat diet. It's really a high-fat, high-sugar diet because the fat is not palatable alone. So they have to lace it with something to make it palatable. They have to increase the salience of the fat, and they do it with sugar. So when you actually give the animal lard with no sugar instead, they don't binge, and they don't show signs of dependence. So fat does not work in rats. In humans, there are plenty of binge foods, and they're high in fat, but they're also high in carbs and sugar, like pizza and ice cream. And if you don't think pizza is high in sugar, take a look at the tomato sauce. Okay, adding sugar increases the preference for fatty foods. That is true. I won't argue that. The point is, if you separate them completely, fat from sugar, the fat does not drive binging. People on the Atkins diet do not show signs of dependence. And lastly, turns out energy density is stronger association with obesity and metabolic syndrome than specifically fat. So, fat, not addictive. Now, caffeine is another story. Caffeine is a model drug of dependence, okay? And if you take my Starbucks away from me, I will kill you, okay? That is my drug of choice, okay? And the day I find out that caffeine is toxic to health, I will blow my brains out. Okay, so I'm just making it very clear. I do have a vested interest. I do have a conflict of interest. Okay, I am a caffeine addict. And I'm not going to say I'm proud of it, but you know, it is what it is. So in humans, dependence has been shown in children, adults. 30% who consume caffeine meet the DSM criteria for dependence, and the physiologic addiction has been established. You get headache, you get impaired task performance, you get fatigue. You know, doctors drink coffee, and they always drink it black. All right, so caffeine, yes, drug of dependence, no argument, caffeine addictive. Which leaves us now with the last one, sugar. So there's the question. This is where we're laying it out. Is sugar, and specifically the molecule fructose, because that's the sweet molecule, is fructose addictive? So they say, Neurofast says, not addictive. Right? They say, not addictive. I'm going to play you now a video that's going to say something else. And I think you'll all recognize who this is very quickly.
I couldn't get through a day without doing something to alter my consciousness.
And it started with heroin, I think.
It started with sugar.
Sugar.
Oh yeah, when I was 5, 6 years old, I was cramming sugar down my throat as fast as I could get it down. Sweets, you know, sugar on bread and butter. I became addicted to sugar because it changed the way I felt.
So there's Eric Clapton talking about how he got into heroin. Sugar is a gateway drug, and it is. And our kids are exposed to it nonstop. And I can prove it. Does anybody know what this is? Anybody ever work in a newborn nursery or a neonatal intensive care unit?
I did.
Okay. So does this look familiar?
Yes.
Okay. This is called Sweeties. Sweeties. It is a super concentrated sucrose solution, 24% sucrose solution, that you dip the pacifier in and then stick in the newborn boy's mouth before the circumcision. Got it? Why? The Jews have wine and everyone else has Sweeties, because it elicits both a dopamine and an opioid response in the nucleus accumbens, even in neonates. Because sugar is addictive, that's why. And I'm not the only one who says so. National Geographic said it, why we can't resist it. And indeed we can't. We cannot resist it, because it is addictive. So, sugar addiction. What we're looking for are similarities to other drugs of dependence, like nicotine, morphine, amphetamine, cocaine, cannabis. The one I think is the big one, the one that to me makes the sense, is alcohol. So, who here drinks alcohol? Anyone? Even socially? No? You're all teetotalers there in Iceland? I don't believe you. 40%. 40% of America are teetotalers. They don't touch this stuff. 40% are social drinkers. They can pick up a glass of wine and put it back down. 20% are hardcore addicted, and 10% are chronic alcoholics. Probably the same with sugar. And it makes sense that that would be true, because after all, where do you get alcohol from? Fermentation of sugar. It's called wine. So the fact that they have metabolic similarities shouldn't be too surprising. So what makes a milkshake so rewarding? So Eric Stice at the University of Oregon, which I believe Nicole talked about, actually stuck people in scanners and tried to answer that question. What they did was they went into the scanner with a straw, a plastic straw that was connected to 2 bottles, one with fat, one with sugar. And so he could modulate the percent of fat and sugar in the milkshake that these people were consuming while they were sitting in the scanner. And what he found was that adding fat did not stimulate the reward system. Only adding sugar did. The fat stimulated the cortex, the somatosensory cortex, mouthfeel, if you will. So, I mean, there was a sensory experience associated with the fat, but it didn't impact the reward system. Only the sugar impacted the reward system. This has been carried out now in Europe as well, where they've shown that glucose and fructose can be dissociated, not just sugar and fat, but glucose and fructose, because it turns out that the glucose stimulates the caudate, the putamen, the basal ganglia, the precuneus, the lingual gyrus, all important, but not the reward center. Fructose, on the other hand, stimulates the reward center. So glucose, the rest of the brain, fructose, reward center. And my good friend Anya Jastreboff at Yale, working with Mark Potenza and Bob Sherwin, basically took this apart even better, showing that fructose doesn't, it basically does its work only in lean people, not in obese people, because they're already downregulated their receptors.
Dr. Lustig, can I just ask you a question right while you have this here? It's
Sure.
just easier. What about something like stevia, which is an artificial sweetener and has I guess a sweet base?
Right.
Where would that fit?
Right. Well, we don't know yet. We do know that stevia does stimulate the reward center. We do know that. We don't know if it's the same, and we don't know if it downregulates receptors. That work has not yet been done. Everybody wants to know about diet sweeteners, me too. And there are a lot of reasons to be concerned about diet sweeteners. But Vera, the question you asked, we just don't have the answer to yet. It's one of the things that has to be done and hasn't been done yet. Okay, we can absolutely talk about diet sweeteners when we're done and whether or not they have any role or any place in this argument. Okay, and I'm happy to discuss it, but I will just sort of give you the punchline. We don't know. We just don't know yet. I'm worried, but we don't know. All right, what about in humans? Now remember, we went from the DSM-4 to the DSM-5 5 years ago. Remember 2013? Now, the DSM-4 very specifically said you needed tolerance and withdrawal to be addictive. Now, withdrawal are systemic effects of abstinence. So for cocaine, you have systemic effects, irritability and jitteriness. For caffeine, you have systemic effects, heart rate, sweatiness, fatigue. For opioids, you have systemic effects. You have GI upset, nausea, etc. Okay? For nicotine, you have systemic effects, dry mouth, jitteriness, etc. All of these are systemic effects, not central effects. But the DSM-5 had to fit gambling, which is not a drug, into the addiction framework in order to be able to provide clinical services. So in order to put a behavior into addiction, they had to change the definition of addiction, because gambling's not a substance. It's an addiction, but it's not a substance. Therefore, you wouldn't expect it to have withdrawal symptoms, because withdrawal symptoms are all systemic effects because of chemicals, not because of behaviors. So they had to change the definition. So the DSM-5 criteria for addiction say tolerance and withdrawal or tolerance and dependence. Here's the list of dependence criteria. So I want you to read through them real quick. I'm not going to read them to you. Just read them yourself. And tell me, does that sound like obesity to you?
Yes. And
Exactly.
yes. And yes.
Yes, yes, and yes. Okay? So obesity is tolerance and dependence, not tolerance and withdrawal. So there's an addiction component here. The question is it eating addiction or is it food addiction? Okay, we're still trying to answer that question, and that's where the definition, the legality has to come in. So let's return now to what NeuroFast said, because I want to now take their argument apart, limb by limb. This is what I showed you at the beginning. In humans, there's no evidence that a specific food ingredient, or food additive causes a substance-based type of addiction. The only known exception is caffeine. So they give caffeine a pass. Then they say, within this context, we specifically point out that we do not consider alcoholic beverages as food, despite the fact that 1 gram of ethanol has an energy density of 7 kcal. Well, in fact, caffeine and alcohol are not food. They are really food additives. They're things you put into food to make them addictive, not food themselves. So my question to you is sugar a food? Yes or no? Is sugar a food?
No.
Well, let's take it apart. Okay, we need some guidance here. I happen to agree with you, but let's go there. So here's the Food, Drug, and Cosmetics Act of 1938. This is, the FDA, the original FDA. This is what they came up with in 1938. The term food means, 1, articles used for food or drink. The first law of vocabulary is you're not allowed to use the word in the definition. What do you think? Throw that out, because that's just bullshit. Let's go to Webster. Here's Webster's definition of food. A material consisting essentially of protein, carbohydrate, and fat used in the body of an organism to sustain growth, repair, and vital processes, and to furnish energy. Okay. Energy. Furnish energy. So, sugar provides only energy. That should make it a food, based on Webster, right? So can you name me an energy source that is not necessary for life, that there's no biochemical reaction in the body that requires it, that is not nutrition by any dietitian's stretch of the imagination? There are 0 dietitians on the planet that would agree that this energy source is food or nutrition, that when consumed in excess, it is toxic to cells, to organs, to humans, and causes death. And we love it anyway, and it's addictive. Anyone? Alcohol. Alcohol is not food. Even NeuroFast says alcohol is not food. Okay, but it's an energy source. It's 7 calories per gram, but they gave it a pass. Why? Because they don't want to deal with it. But alcohol is not a food. It's an energy source that's not a food. So just because you're an energy source doesn't make you a food. So Webster's is wrong too. So the FDCA is wrong and Webster's is wrong because no one knows how to define food. In this article, I delineated the metabolic similarities between fructose and ethanol, the hedonic similarities between fructose and ethanol, and how society treats both of these, which is virtually identically as well. And it makes sense that that would be the case. So aside from sugar, are there any other energy sources that are not food? And the answer is alcohol and trans fats. Trans fats used to be food, now they're not. We took them off the generally recognized as safe list in 2013. They were reclassified because we learned that they are consumable poison. Okay, they're an energy source, but they're not food anymore. So my question is, should sugar be a food anymore? The FDA has reclassified trans fats and nitrates as not generally recognized as safe. Nitrates cause cancer, colon cancer. could we reclassify sugar as a food additive instead of a food? Because after all, we call it added sugar. Because it's a food additive, added sugar. I'm not worried about the sugar in fruit. I'm worried about the sugar in Coke. I'm also worried about the sugar in fruit juice, because when you take the fiber out, it is a Coke. So could we get sugar reclassified as a food additive? Well, addiction doesn't get you off the GRAS list, because caffeine's on the GRAS list. So here's the definition of GRAS, just generally recognized as safe. Generally recognized among experts qualified by scientific training and experience to evaluate its safety, that would be me, as having been adequately shown through scientific procedures, or in the case of a substance prior to January 1st, 1958, which sugar fits into, either through procedures or experience based on common use. That's where sugar comes in, common use, to be safe under the conditions of its intended use. In other words, sugar was around in 1958, and we put it in 5-pound bags and sold it at the grocery store. So therefore, it was safe under its conditions of intended use. Everybody remember 1 lump or 2? Well, now the 2 lumps are in everything. That was not intended. That was not its intended use. And that postdates 1958. GRAS is about toxicity, not addiction. The problem is our current consumption was never intended. Now, sugar's been known for thousands of years. The original sugar cane extract, which they could put into pottery earthenware and put on ships out of India, had a name. It was called khanda, K-H-A-N-D-A, the derivation of our current word candy. Okay? It goes back a long way, but we never had very much of it. It was rare. And I think you know through the Middle Ages, sugar was fetishized. They used to make sugar sculptures in palaces. The kings would commission sugar sculptures. And, to this day, the pastry chefs, love making those sugar sculptures. You've seen them. The last time the FDA looked at sugar was in 1986, and that was before the high fructose corn syrup glut. And they only had data up to 1980, which did not include high fructose corn syrup. So the average consumption at that point was 51 grams per day. And that's half as much as we consume today. So the question is, if we did that evaluation today, would the data be different? And the answer is absolutely. And it would show toxicity at our current use, because our current use was never intended. So you're probably all familiar with this New York Times Magazine article about our research at UCSF, which Gary Taubes wrote, called Is Sugar Toxic? And here's our comment that my colleagues Laura Schmidt and Claire Brindis at UCSF and I wrote in Nature, called The Toxic Truth About Sugar. So I pose to you, you know this on the left. This is, shall we say, bringing coals to Newcastle for an addiction group. I would also propose this is true as well.
Agreed, yes.
So this is what's happened to our
Yes.
food. This is what's happened to our food over the past 30 years. 1982 to 2012. Meats, down 10% because we were all told to go low fat. Now, fruits and vegetables, you'll notice exactly the same. No change. Now, we're always told we don't eat enough fruits and vegetables, and that is absolutely true. We don't. But we never did. Ain't new. And we didn't have this problem in 1982, but we sure as hell have it now. Because it's not about the fruits and the vegetables. It never was. Okay? This is a subterfuge on the part of the food industry to take our eyes off the problem so that they can assuage their culpability. Well, we don't eat enough fruits and vegetables. Total, complete, utter bullshit. Grains and baked goods. up 1%. And that's important because grains and baked goods is refined carbohydrate, which is glucose, glucose without fiber. Glucose raises insulin. Insulin means weight gain, as we've talked about. It's the energy storage hormone. So the more refined carbohydrate, the more weight gain. So this is part of the obesity equation for sure. I'm not arguing that. But what I'm talking about is addiction and metabolic syndrome. And that's not where this is. Dairy products, down 2.5%, because now we're all lactose intolerant, right? So if you're lactose intolerant and you don't drink milk, what do you drink instead? You got to drink something. Okay, if you drink water, you're good. If you drink anything else, not so good. And finally, processed foods and sweets, 11.6, 22.9%, a doubling in those 30 years. This is the problem. This is the problem. And the reason that NeuroFast calls it eating addiction is because all the food is now laced with sugar. So all the food is addictive because it's the sugar that's addictive. So they can't tell the difference between food addiction and eating addiction because there's nothing to compare it to, because all the food has it. Of the 600,000 items in the American food supply, 74% of them have been spiked with added sugar. And more than 50% of the sugar in our diet is in ultra-processed foods. So, in summary, it's not about obesity. Obesity is a marker for the problem. It's not the problem. It's a result of the problem. When you see somebody who's obese, that's sad. But you know what? The normal weight person standing right next to him is just as sick and just as addicted. Food addiction is a misnomer. So I agree with NeuroFast that it's not food addiction. But I disagree with them that it is eating addiction. Because really what it is it's food additive addiction. And if we talk about food additive addiction, and specifically sugar and caffeine addiction, which are the only items that are placed into food specifically to get you to consume more, then it all makes sense. So do not talk about food addiction. Talk about food additive addiction. So the only junk food that is actually addictive are sugar and caffeine, and they are food additives. Fat and salt increase the salience of those foods, but they themselves are not addictive. They're like menthol in cigarettes. They increase the salience, but they're not themselves addictive. Just because something has calories doesn't make it a food, even though the FDCA and Webster's would argue with you. But logically, that just doesn't work. Okay? It can be a food additive, though, like ethanol and trans fats are food additives, not foods. The only way to fix this problem is to take sugar off the GRAS list. Because as long as sugar is on the GRAS list, the food industry has carte blanche. Because after all, if it's generally recognized as safe, that means it's generally recognized as safe at any level, which means they have no reason to want to cut back. Because sugar is cheap and it takes up space. My favorite, my way of explaining this to you, I don't know if you have this in Iceland, but here in America, we have a breakfast cereal called Lucky Charms. Do you have Lucky Charms in Iceland?
We
Yes,
sure
no?
do.
Huh?
We sure do.
You do, okay. Everybody familiar with Lucky Charms, right? Orange stars, green clovers, yellow moons, pink hearts. Why are there marshmallows in the box? Why are there marshmallows in the box?
So they're going to buy and eat more.
Well, yes.
But there's something else.
But Frosted Flakes will get you to buy and eat more, but they don't have marshmallows. Why are there marshmallows in the box? Reason? Because oats are expensive and marshmallows aren't. They take up room in the box. They take up space in the box. So they can put less money in the box and sell it for more. Great business strategy.
That's a good pointer.
Okay? So the only way to fix this problem is to get sugar off the GRAS list. Otherwise, the food industry will continue to use it with impunity. Now, the problem with GRAS is it focuses on toxicity, not addiction. Now, it just so happens fructose is both addictive and toxic. And sugar is toxic in its current dosage, which was never intended. So if we revisited this argument and looked at the data today, as opposed to the data from 1980, we would get there. The problem, of course, is that the FDA is under no obligation to do this unless directed by the president. Well, our president has already taken the new food label and deep-sixed it. So we won't even know how much added sugar is in food. And the reason is because he wants the food industry to sell because he's all about the money. So this is not going to get fixed, at least in America, in the next 2 years. But what we can do is we can be providing the bench work, the ground game, so that when we do get a new administration, we're ready to go. And my job, my unwritten, my self-appointed job, is to try to ally the medical profession, the dental profession, and the dietary profession around this issue, so that when we do get an administration, we are organized and ready to go. Now, I'll tell you, the dentists are on board. The doctors are coming on board. The dietitians, they suck because they are paid off by big food. They will not change because for them, it's all about calories. Okay? This is not about calories. None of this has anything to do with calories. The reason they're all about calories.
One dietitian who believes in food addiction.
Okay, well, good. I'm glad to hear it. But as you know, if you're a dietitian, the first thing you learn in dietary school is about calories. And I know that my cousin's a dietitian. And the reason that they learn about calories is because calories is math. We're talking science. Okay, not everybody can do science, but everyone can do math. So with that, I want to close and thank all of my colleagues at UCSF, at Touro University, at San Francisco General, my colleagues at the Institute for Health Policy Studies, Nicole, who has been very helpful in terms of our discussions to try to formulate this all together. I am a big, big fan of Nicole's. We both agree on this issue, and I want you to start calling it food additive addiction rather than food addiction, so that people will get the picture. And that's not just semantic, because what you're doing is you're making the argument that it's not the food itself, it's what's been done to the food. All food is inherently good. It's what we do to the food that's not. Okay?
Thank you very, very much. And I think those words are really well spoken. And I know for me and us in the field of food addiction, as we've called it, we've multiple times we come back and we go back and forth to how to explain it in the best of ways. And I think you put up a great argument for the for even changing it further, or at least indicating that as some part of it. But one question from me here in Iceland is as we go forward with all of this, where do you see the food addict or food addiction professionals come into this alliance of yours as we go?
Absolutely. Esther, you have to lead the charge. Okay? I mean, without question. I mean, you are the ones who make the case, okay? Because you're the ones in the trenches.
Right.
We need everybody. We will need everyone. Here's the thing, okay? Think about societal interventions for public health crises, okay?
Yeah.
Every public health crisis has required personal intervention, which we can call rehab, and societal intervention, which we can call laws. So nicotine, alcohol, street drugs, cocaine, heroin, pick your addictive substance. Rehab and laws, rehab and laws. Personal intervention, societal intervention. Do you think you can mitigate any addictive pathology if you don't have both? You need both. You can't do it one or the other. You have to have both. So professionals like yourselves can be working on the rehab side, the personal intervention side, because you're dealing directly with the victim, the patient. And by the way, they are victims. They are not perpetrators. No one chooses to be addicted.
Right.
There's not a person on the planet who chooses to be addicted to anything if they had a choice. But the problem is, once you're addicted, and the problem is, if you don't know that you're getting addicted, how would you stop it? You know, everyone knows cocaine is addictive. Everyone knows heroin is addictive, okay? But no one knows that sugar's addictive, and no one knows what the sugar's in. So people are being addicted and don't even understand it, don't even know it. They think when they, you know, order a salad at McDonald's that they're doing better. Except, have you looked at the dressing? Yes. So I mean, so you have to treat them as victims, not perpetrators, which of course you all do because you're because you're addiction specialists, so you get it. But the rest of society does not. And that's where you can come in, is helping the rest of the public recognize what the real problem is and how to deal with it. And it will require a societal intervention.
Yeah.
Vera, you had a question.
I do, thank you. First of all, I want to say thank you so much. There was some repetition from when you spoke, but you really got more into the science, which I personally really appreciated. So thank you so much for your talk. It just clarified things even more. I want to go back to my question about the sweetener, and I'm specifically asking about stevia because that's the one that's considered the healthiest of all the artificial because it's a natural sweetener. So I don't know if you have more to say other than we don't know, but could you comment on the fact of artificial versus natural artificial sweeteners?
Sure.
Yeah, whatever you.
I'm happy to tell you what we know, but unfortunately it's not much. Okay, so there are four, count them, 4 reasons to be concerned about artificial sweeteners as a whole. I will tell you that stevia is probably the safest of all the artificial sweeteners. That doesn't mean it's safe. Okay, it is probably the safest of, but that doesn't make it safe. So here's the issue, okay? And there are 4 of them, but we don't have the data for any of them for sure. They're all question marks at the moment, but I'll give you all 4. Number 1, there's this thing called pharmacokinetics. There's this thing called pharmacodynamics. They are not the same. Pharmacokinetics is what your body does to a drug. Pharmacodynamics is what a drug does to your body. They're not the same. Now we have all the pharmacokinetics for all the diet sweeteners because the FDA demands it. That's in the FDCA of 1938. Because pharmacokinetics tells you about acute toxicity, whether you keel over and die. So we have acute toxins like sarin, ricin, VX, cyanide, you know, parts per billion, keel over and die on the spot. Okay, we make secret agent movies about them. All right, everybody with me? We have no problem calling those toxins. But then we also have things like carbon tetrachloride, benzene, arsenic, tobacco smoke, None of these are acutely toxic, but they're all chronically toxic. They won't kill you from one exposure, but they will kill you eventually. Chronic toxins are not regulated by the FDA because that's PK, that's PD, pharmacodynamics, what a drug does to your body. So will one cigarette kill you? No, but will 10,000 over 10 years? Absolutely. Okay. But the FDA does not regulate chronic toxins. It's not in their charter. So the food industry goes, hey, we have this sweetener. The FDA has approved it for use. Doing these pharmacodynamic experiments take years, take thousands of patients. No one's asking for it. Compounds already on the market. Hey, we're not going to do them. And then the NIH says, hey, food industry's compound, food industry's drug, food industry's profit. We're not going to pay for it. So you got an FDA who doesn't ask for it, you got a food industry who won't do it, and you got an NIH who won't pay for it. No data. That's problem number one. Problem number two, you put something sweet on the tongue. What happens? Message goes tongue to brain, sugar bolus is coming. Message goes brain to pancreas. Sugar bolus is coming, get ready to release the insulin. Well, the insulin vesicles in the pancreas are standing there at the membrane, ready to be released, ready to explode out into the periphery to then store energy. But it was a diet sweetener. What does the pancreas do? Does the pancreas go, oh man, I was waiting for that. I guess I'll just wait till tomorrow. Or does the pancreas go, I got all these insulin vesicles sitting here on my membrane waiting to be released. I'm going to go find me some calories to work on. And you end up overeating to cover the insulin rise that you will get because your pancreas has been stimulated by your vagus nerve. Which is it? We don't know. There are early data to suggest the latter, that you will end up overeating. And there are a couple of acute studies that suggest that. And I can give you the references if you want. But we don't have any chronic studies. We don't have any long-term studies to say that that actually is a driver of weight gain. We have correlation, but not causation. So that's problem number two. Problem number three. Elenov's group, Aaron Siegel and Elenov at the Weizmann Institute in Israel showed in a paper in Nature a couple of years ago that sucralose and aspartame both alter the gut microbiome and cause leaky gut and cause glucose intolerance. And if that's true in humans, which we don't know yet, but if it is true in humans, then that would mean that diet sweeteners, or at least specific diet sweeteners, I don't know if all of them, but specific diet sweeteners could actually be harmful all by themselves, having nothing to do with calories, because they affect the gut microbiome and because they affect gut permeability. And so we don't know that yet either. And then number four is something new. I don't know if you can read it, but this came out this is in Clinical Endocrine News about a month ago, and I'll try to put it where you can see it. It says sucralose may, what does it say? Sucralose may upregulate adipogenic genes in high BMI. Okay, so this is done in vitro with adipocytes in vitro, throwing sucralose in the dish with the fat cells, and the genes that drive fat cell synthesis go up. If that's true in humans, in vivo, then maybe the energy storage is going up irrelevant to the calories, in which case things would just get worse. So we don't know that yet. We don't know that's true in vivo. That's true, it's only in vitro, and it's only for sucralose. We haven't looked at any of the others. We don't know if stevia would do it too. There are all these questions about what these compounds do unrelated to calories, okay? Because they're chemicals, and anything that's a chemical might have other effects, and we just don't know yet. Am I worried about it? Absolutely. Am I ready to talk about it in public? Absolutely not. Because I'm about the science, I'm about the data. You know, the only reason I'm still standing is because I have credibility, because I don't go past the science, right? We just don't have the science for this yet, as much as I would like it. And someday when we do have the science, I'll be very happy to talk about it. But
That's
we don't.
great. Thank you. Thank you. It's wonderful.
So that's where we are.
May I ask a question?
Of course.
Hi, Robert Lustig. This is Bitten Johnson from Sweden.
Pleasure.
I have been working with this for many years. And is it okay to call it sugar addiction for now
Sure.
instead of sugar additive addiction?
Sure. No, I mean,
Okay.
So food additive addiction and added sugar being a food additive. So it's fine to call it sugar addiction.
Good, because that's what I've done.
The only two items I know of in food that are addictive are sugar and caffeine.
Great. Okay, then I understood you right. But I have worked on developing a screening instrument and a diagnostic instrument to be by ICD-10. And looking at your DSM-5 saying that there is no withdrawal with sugar, is that what you're saying?
There's no withdrawal, right?
Well, I tell you what, I have worked with thousands of clients for the last 18 years.
Yep.
And there is withdrawal like crazy. So, you know, do you mean that it is not in any science, or because empirical, I would tell you that if you do cold turkey, people have tremendous withdrawal.
Okay, so question. Is it sugared beverage consumption that you're cutting?
No, I cut all sugar and all flour and all artificial sugar, everything.
So you may be right. I'm not saying you're wrong. The problem is that most of the data thus far, looking at this question, has been in cutting sugared beverages, because that's the plurality of added sugar consumption is in beverages. The problem there is that sugar beverages are both sugar and caffeine.
Yeah.
And it's hard to dissociate the withdrawal, which they definitely get. There's no question. If you stop soda, you get withdrawal. I totally agree. But is it the sugar withdrawal, or is it the caffeine withdrawal, or is it both?
Or is it is it keto flu? You know, if you go into like an Atkins diet from day one, these clients have severe withdrawal for at least 10 days.
Yeah, well, there's no question. We took the sugar out of kids' diets and substituted starch. You know, 9 days of isocaloric glucose for fructose exchange. We've written 3 papers. We're about to submit our 4th on Monday. And so I can tell you, because we did this with 43 kids, for the first 5 days, they were bouncing off walls. They were very unhappy, and they were very irritable. And the parents were really upset. And we told them, you know, just wait it out. And then it was like the fog lifted.
It's just,
and the parents noticed that their behaviors got better. They were less irritable. They were able to study better. They were, you know, cognitively more functional in school. The teachers noticed it. They were less disruptive in class, you know, and the parents, they got it. They saw it. But
no foggy brain. Yeah.
Right. Yeah. Right. They had to unfog the brain. And so, but we did not build an instrument into that study to look at that question, unfortunately. And to be honest with you, even if we had built an instrument into that study, we would still be left with the question, was that the sugar withdrawal or was that the caffeine withdrawal? Because a lot of them were in sodas. So you might be right, Ms. Johnson, and I'm not telling you you're wrong. I'm just telling you that the science is having a hard time differentiating those two phenomena.
Okay. Okay, and I also want to thank you so much for everything you've said, because I love it, and I'm going to fight this tooth and nail all the time. And it is a gateway drug. I'm a recovering alcoholic too, and I think the opiate crisis is due to sugar in the U.S.
Well, I'm not going to argue with you. If you read my latest book, I think you will learn a lot.
I have it. Yeah, I just loaded it down. Yeah, thank you.
Hacking of the American mind. It will make the pathway very clear.
Yeah. Thank you.
Can I ask a question here? My name is Teresa.
I will just tell you, I only have 3 more minutes because I have to drive my daughter to a rehearsal for the opera.
How wonderful. Thank you for this wonderful program. But I've documented 31 days of withdrawal in my clients when they put down sugar. And the second thing I have to say is that in my grocery store, the ingredient list on stevia that's for a pure stevia package says pure stevia on the front and dextrose, stevia on the back. So we have to be careful to get pure stevia.
Okay. I won't argue that.
But thank you.
There's a lot of subterfuge. It's complicated.
And Robert, I know you've been here longer than we had expected, and really generous with your time and your energy. I know that I speak to everyone here that we really sincerely appreciate you, your work, and where you're coming from, and hope to see you again and again and again here.
I hope so. Happy to help, Esther.
Thank you so much.
Thanks very much for attending, everyone. All right. Have a good rest of day.
You too.
You too. Bye-bye.
Thank you for being here.
Pleasure.
Okay, so I'm just going to stop this here.
Great work, Esther.
Yes, this was wonderful, wasn't it? Oh my God.
Wonderful.
Yeah. Thank you, everybody, for being here and with us and enjoying this. I think I agree with what he was saying, and what I I think is so essential for him is not to go out of the science part. If it has science, it is there. Just you guys take a look at it, or be stupid. But what we need to do is add to science, add to the science. That's what is lacking, I think, for our profession. That is just in the, we're just green. We're green. We're in the green. And so I am, what I've been thinking about very much in the past few weeks is what can we really do through the school, through the teachers, through the students. There are, incredible professionals coming out of this Impact School and other, is one of which is to continue and advance Bitten's work on screening and mapping and assessing the diagnostic tools and setting up clinical guidelines for professionals, for us to begin with, and then for others, as people will start slowly but surely want what we have and want the knowledge that we have and the experience that we have. And hopefully we will get more scientists come in, or people doing more research as we go on. And I'm optimistic about that. So that's my 2 cents on this, and I'm going to turn off the recording, but we still need to stay on a little bit, and I will